opportunity table and plants for sale. And Jim Heilig's here with us, so Adam will introduce him in a bit. And then should we go to the announcement slides?
Yeah. Sure. Welcome, everyone. Hope you can all hear us. We're going to go through some announcements, then we'll pass it over to Jim. He's going to talk about genetic and breeding concepts for orchids. Then, of course, we'll have our show and tell, both in the room and on Zoom. Then we've got opportunity tables for those of us who are here in the room. And we have the gift certificate raffle to Sunset Valley Orchids for online and in-person attendance. We are, of course, recording. And here we go. First announcement, most exciting one, is we have one new member this month. So welcome, Natalia Bourne. We're glad to have you. I believe she's online.
Welcome.
So welcome. Of course, there's judging. We've got judging here in the other room tonight. And there is also judging down at Allele House on the 15th. And then up in Sacramento, I believe it's on October 4th. The details on the screen. So check that out. Events here with us. As you know, we are already planning for the 70th annual Orchid Pacific Orchid Expo. It's going to be the end of February. Marie Brar has volunteered to be our show committee chair, so we're very grateful for her efforts in the room. Thank you so much. And of course, there's lots of other ways you can volunteer and get involved before and during the show. So please reach out and get involved. There'll be a show committee next Monday if you want to get involved that way. We've already got exhibitors signing up. There's a floor plan going. Like it's already happening, just several months away. But at the last board and show meeting, we picked a theme. Out of all the entries, the voting decided on celebrating diversity. So we're excited about that. We've got a designer, Jonathan Fong, who is already hard at work on a poster. He's given us his 2 current drafts. These are drafts, so you know they can change. They're subject to change. Don't you know? But anyway, here they are. All right.
He's still working on the background colors. And other things.
Yeah, things will tweak the next time you see it, but it's already off to a nice start.
And then, yeah, we have vendors coming. We haven't sent the exhibitor invites out yet.
Oh, man. Okay.
So for the different societies, we'll be sending those out for individuals that want to do exhibits. And the show committee meeting next Monday night is going to be hosted online as well as in person at Nishi's Hair Salon in Dog Pound. So information, if you're interested and didn't get on the invite, let me know. We'll get you on the invite. And Maria's in the back of the room, show chair, so introduce yourself and get in the back of the room.
Oh, next month's speaker is going to be Fred Clark from Sunset Valley Orchids. He's coming out and he's going to give a presentation on becoming an 80th percentile grower. Okay, good. He said it was good. I thought it sounded fun. So we're looking forward to seeing him. And then he's going to head up to Sacramento the next night. So if you want to double jokes, you know, you can caravan. We mentioned this last time. We do have a survey that we're going to send out to get your thoughts on what you'd like to see in programming for the next year, the types of activities we have, the types of content. So we'll be sending that out.
It's actually on the members section of the website. It's already been inside of the members section, and there's going to be a link on this main public page as well that you can access. So if you missed the email, you can just go to the website and click on it and do it.
Yeah, let us know what you're looking for.
And to give her appreciation, Lynn, or did you put this in there? So Lynn is stepping down from doing the virtual show and tell after this month. So I mentioned this not only to acknowledge and thank her for all her hard work, but we're going to look for a new person to change the paradigm. Here's John. For those who don't know Jonathan, that was the artwork that you saw. Was Jonathan? So we're going to be shifting again. She's done it's been a couple of years now, I think, a year or two. So part of that survey is input on what you want to see for skill sessions, general programming, as well as how we do show and tell hybrid. So we want that input from everyone. So please click the link, look for the email, go on the website, fill it out. You can do it anonymously or with your name.
Jeff, is this a different survey than the one I did 3 or 4 weeks ago?
It's probably the same one, but
Well,
we added.
There wasn't anything about show and tell.
Yeah, because we just found out. So we were just talking to Lynn before the meeting. So it's now, there's additional questions. So if you want to do it, just go in again. And
It's one additional question.
you can go back in and change your answers or bad answers. But
yeah. Sorry. Other events that aren't ours, the California
cards.
they're hosting Panning for Orchid show up in Sacramento at the end of this month. We've got cards here, but you can also see the link to go and get more information. Orchid Digest has a virtual event on Saturday, November 12th. You can find out more information on their website. And that's what I have for announcements this month. Oh, here's the card that we have in the room. Let me take a quick look here before I introduce Jim. We have chats. People are sharing the link and thanking everyone for Lynn. Thanking Lynn. Great.
Thank you for the kind words, everybody. Thank you.
Yes, we'll all appreciate it. And we will miss you.
Oh, I know the other announcements. So next month, you mentioned there'll be Fred Clark, but for December, it's going to be no online, it's just in person. In the auditorium, we're going to do just a mixer holiday party with potluck. So no speaker, so there won't be an online part in December.
Yeah, but anyone who can come in, we look forward to seeing you. Great. Well, I'm going to introduce our speaker, Jim, here. Some of you probably already know him, but he's been growing orchids for over 20 years. Over that time, his interests have revolved around Phalaenopsis and other Vandation species, along with many other genera. He's been breeding orchids for over 10 years, with particular interest in exploring the genetics of various Phalaenopsis species in simple hybrids, primary and near primary, with a goal of bringing fragrance back to standard hybrids that have the grace and ferociousness of the species in their background. More recently, he was focused on propagating species while dabbling with a few hybrids. While Jim has grown under lights for many years, he was discussing this earlier in his electricity bill, he is currently exploring species and hybrids that can be grown outdoors in his coastal Monterey Bay area garden. Those requiring cozier environments are grown in a small greenhouse. He began his judging career at the Great Lakes Judging Center out in Ann Arbor, Michigan, and is currently a judge, for now, at the Pacific Central Judging Center. He has held several leadership positions at the Orchid Society, the AOS, and at the judging centers. In his day job, Jim works as a raspberry breeder, as those of us here in the room have discovered. His focus is on pre-breeding, which involves discovering novel traits in the wild and unadapted germplasm and introgressing those traits into adaptive backgrounds to improve disease resistance and flavor, among other traits. We've got some very interesting looking ones here. But for his actual talk tonight, he's going to discuss basic genetic and breeding concepts as they pertain to orchids. He will share his experiences and observations on some of his work to explain these concepts with examples from his breeding program. Having a better understanding of these concepts can help those interested in preserving species or growing hybrids to make informed decisions on acquiring plants for their collection or to propagate. With that, welcome, Jim.
Thank you, Adam. Hey, before I get going, can everybody hear me online? And is it coming through okay? Is it?
Yes, just
I like
fine.
having the door open, but there's a lot of traffic noise. I'm wondering if we should open the back one instead, or do you want to just leave this one?
Just open the hallway door for ventilation. Just close this one.
I'll go ahead and share my screen here. Thank you. Okay. First time I can actually check to make sure that everybody's seeing it myself. So thank you for the introduction, Adam, and thank you for inviting me to talk to your group tonight. I'm always excited about the San Francisco Orchid Society. I see so much activity and so much volunteer and good programs here. So I feel honored to be asked to come and talk to you. So as Adam mentioned, in my day job, I am a plant breeder. So I deal with a lot of these concepts on a daily basis. And I do bring it into my orchid hobby. Actually, I was breeding my orchid hobby before I went into plant breeding as a career. I hadn't thought about being a plant breeder for a while. I actually did my undergrad and worked for a while before I went back and said, hey, I want to be a plant breeder. So the orchid breeding actually came first. So it's also then challenging to put in here. So I have a few disclaimers at the beginning, but also I know there's a huge range of differences in experience in plant breeding. I know there's people in here probably that do process themselves or propagate species, to people that don't and are just enjoying the orchids in a collection and don't have any interest in doing any crossing. But most of us are buying plants, I imagine. Who hasn't bought a plant in the last 2 months?
Last 10 minutes.
10 minutes. I traveled for a while and it was a joke. My husband said he missed me, but every other morning a box arrived at the door with orchids, so he thought of me and I told him that was the plan. But so I chose topics. I hope it's not too boring for some, and I hope it's not too over the top for others. So if you have a question, we can please stop, get my attention, we can talk about it. I intentionally didn't go very deep into a lot of these subjects because some of these subjects would actually be a whole semester at a university. So for various reasons, it's going to be a little light. So said at first, we're going to kind of do some disclaimers, and that is an overview. Many of these concepts are very deep, and you can go online and find lots of information that goes even deeper. There's lots of good articles out there. Be careful of forums. I'm not a member of any forums at the moment, and I know they're very popular, but a lot of times I see misinformation on them, especially about breeding and then things like that. So just always take those things, I say with a grain of salt. Somebody said with a salt block of salt. So keep that in mind. The other thing, sometimes things don't follow the rules, quote unquote rules. And so concepts like that I might talk, you may have a different experience, and there might be a very good reason why. And it's interesting always as a breeder to talk about those things and when things were different, worked out differently. But be careful of anecdotes. A lot of times when people are talking about like the heritability of this trait, you really need to grow out like 100 or 1,000 plants to actually understand the true habits, characteristics of the genes. And most of us are blooming out 30 or fewer plants. I mean, if you grow out 5 plants and you see something, that doesn't necessarily mean that that's representing what it could have. Part of that reason is the generation time is really long for orchids, so they're not a very good model plant. Most scientists that study like really, genetics are studying a Arabidopsis that blooms in 20 days, and they can have 100 cycles a year to study these things. And the orchids, on the other hand, I don't know if I've ever bloomed an orchid in less than a year from seed. Some do, but not really enough consistently to do a lot of the work to understand orchid genetics. Oh, I also say I'm not a biochemist. So we do a little bit about biochemistry. I kind of gloss over it a little bit. If there's a biochemist here, cool. I gave a talk a couple months ago, and they're about pronunciation of orchid names, and there are actually people that like studied those things in the group. So there were definitely people that maybe had a little bit more background than even I did. So if you do go ahead and if you feel the need to help out, help out. There are some assumptions. I'm gonna make some assumptions that everybody has some level of like education in either high school or college about like genetics and DNA. I'm not gonna go into all that stuff. So there's some of these assumptions I'm going into that each individual has one genome inherited from each parent, just like people do. You know, you get one from your mom and one from your dad. Plants are no different. There's other two concepts, gene and allele. People use these interchangeably, but they're actually not the same thing, and they reply to very different things. A lot of times you'll hear people say, oh, the blue gene for Ketleya, or the blue gene. Well, that's not exactly accurate. A gene is a location in the genome that codes for a protein that makes a trait. The allele is the version of that gene. So you say like there might be a blue gene, but there's actually a pink gene there too. The gene is for the color in Cattleya's, but there's different versions of that gene that make it blue or pink. The other thing is this heterozygous / homozygous. It's a little more complicated, but if you have the same gene from each parent, it's homozygous, homo same. And if you have a different copy from each parent, it's heterozygous, different, hetero different. And so we'll talk about that. It's not very deep, but there are some examples in here. And just the basic kind of dogma that DNA makes proteins. These proteins interact to make a feed, the trait we see, the traits we see with our eyes, also known as the phenotype. And I know another thing, I just put this in here, kind of head it off, because when I've talked about genetics in the past, people start going on, maybe not all the DNA in a cell, in our bodies, for people too, and for plants, is in our nucleus. And that's usually when we're talking about heritability, is to treat the genes in the nucleus. But there are other parts of cells, the organelles like the mitochondria and the chloroplast, specifically for plants, that also have their own DNA. We're not going to talk about that, and that doesn't really have a huge impact on orchid genetics. So I'm going to kind of start out here at the beginning, a seedling versus a clone. I hope everybody knows the difference. And when you see a clone, it's vegetatively propagated, it's asexual, so it's actually an exact copy of the plant that it was propagated from. You know what you're going to get usually, and there's certain times I buy clones because I want a specific trait and I want to know it has it. Whereas if you buy a seedling, it's kind of a gamble. You don't know, they're all individually unique individuals, just like us. But also clones, anybody can get one. They're not necessarily special. You see them at Kroger, or I guess for California Safeway. They're not very special. Mostly what we're going to talk about will apply to seedlings tonight. And these are, just like anything else, they're unique. Nobody else has one. Just like you're different from your sibling unless you have a identical twin. Genetically, we're all different. It's a bit of a gamble. You might get bad, you might get good seedlings, but if you consider the appearance, you can kind of hedge your bets. I personally grow a lot of seedlings just because I like to see the variation, and I kind of like having something that nobody else has. I don't necessarily grow my orchids. I'm a judge, but I don't necessarily buy orchids with the idea that they're going to be judged. I grow them for myself. They happen to get an award, that's great. But I like seeing that variation. And I usually, when I buy seedlings, I usually try to get a minimum of 5 of any seedling. Up to more if I can. So everything we're going to talk about tonight really applies to seedlings. Clones, I have a little asterisk there. Sometimes there's some variation in clones, but that's like a different topic, and we'll leave it there. So I'll start off with kind of when you buy your orchid seedling, a lot of times you'll see like Neopathia Superior Eye by self or by SIB or outcross. So I'm just going to kind of talk about what those different things mean. And a lot of times when people, especially if you're propagating species, how many people are species fans? Like your collection's mostly species? There's a few. My collection's mixed. I enjoy species, but I enjoy hybrids too. I'm a plant breeder, so I have to have some hybrids at least. But a lot of times when people are propagating species, especially, they avoid selfing because they think that selfing is its form of inbreeding. It's kind of the most highly inbreeding situation you can be in. We're actually selfing, taking pollen from the same plant, putting it on that plant. And inbreeding is generally considered to be bad. And that's not always true. Sometimes you only have one plant. So if you want to propagate it in its species, you kind of have to self it unless you can find another clone. And sometimes that's very difficult to do. So people tend to look down on selfing, especially from the conservation standpoint, because you're losing diversity. And I kind of, here's a graph of how fast. So this is the percentage homozygous. So this means that you have the same gene at every location. You see, as you self, you very quickly, at the self, if you self, say 6 times in 6 6 rows, like take one plant, take one of those plants from that selfing, self it again, you'd be almost 100% homozygous by the self-6 generation or self-7. It never gets to 100%, but it gets close. And so you do lose diversity pretty quickly, but a lot of plants can actually handle that for quite a while. And in many of our orchids, I think actually probably have gone through, especially a few species, gone through many cycles of selfing just because of the availability of plants to start with. It also quickly reveals recessive alleles. These are alleles, versions of genes that you don't normally see, but you know, the Flavobessi, the first ones were all orange or the red, but until people started selfing them and breeding a little closer, did we start seeing the yellow ones. So those special kind of unique situations don't come out until you do some inbreeding. You can use it to fix traits. That means that when you cross like a yellow with a yellow, for Flavobessi, you should get all yellows because there's only the variation is gone. Some groups are really tolerant of inbreeding and others aren't. And orchids have done a lot of evolution to encourage outcrossing, the lips, the structure, the flower, fragrances. Most plants have evolved to try to cross to another plant. But I also feel that in my own experience, most orchids can actually tolerate a good level of selfing too. So for a couple of generations, you won't have very significant problems. And there are some orchids that have evolved to be only selfing. Epipactis hellebore, that weedy thing that's everywhere. Who knows what its pollinator is? I mean, it's from Europe, but every single flower sets seed, and they seem to have no problem conquering North America. And there's a few other Oceoclides maculata, an African orchid that's basically taken over much of the Americas, is similarly self-pollinating. So, selfing, a lot of times when I talk to people, especially that focus on species that don't want to self or they don't want to self a plant because they don't want to cause problems, you can do it for a little while, definitely, without any problems and generate more plants. The next so that's the most intense inbreeding situation. The next is doing like a sib crossing or sibling cross, where you actually take siblings from that same cross. You cross 2 plants and you grow out 38 plants from that cross seedlings, and then you cross 2 of them again. I have the graph here. You can see it takes a lot longer to get to close to 100% homozygosity. You have to do that 14 times or 14 cycles to get to the same equivalent as in the F6 for selfing. So you definitely do retain some diversity, the heterozygosity, a little bit longer. You can still reveal recessive alleles because if one of the parents had a recessive allele, the progeny probably do too. So if you cross enough siblings together, you might start seeing those recessive alleles expressed. Similarly, if you cross 2, if you have 2 like alba siblings, alba is generally a recessive trait, you can fix that and produce more albas. And some people kind of refer to this as line breeding. You've maybe heard people say my line or because line breeding something. This is part of that process. So outcrossing is absolutely the opposite of inbreeding, and that's literally where you're trying to find a plant that's not related to the plant you have. And this is really, really challenging when you actually start to think about it, because what does that mean? If you go and I buy a plant, if you buy a plant from I won't use names because, but like orchid nursery A, and you find another one at orchid nursery B, there's a good chance that those actually came from the same source with the way things go. So what do you do if you really want to do an outcross? Do you find I tend to go to maybe some of the foreign vendors, hoping that they're bringing it in from a different source, and that maybe there's like an American, North American kind of gene pool for one species. And many years ago, I decided I wanted to do a really good outcross with Arangus fasciosa, one of my favorite plants, and it seemed like there was a lot of inbreeding, and it was really hard to find something that wasn't related to stuff that was already circulating. As far as genetic diversity, diversity of the genes or the alleles in the plants, this is probably the best, but you have to be careful because if it's a plant from the same population, some plants have really, really gigantic distributions, like Arangus mysticidii and Arangus, there's hybrid Arangus over there. Some of them, they go everywhere from South Africa all the way up the eastern coast of Africa. So if you take one from like Zambia and cross it to one by South Africa, are you making a hybrid? For the registration purposes, we consider that still a species, but some people might think that's degrading the genetics I think at the end of the day, as long as the plants are surviving in cultivation, if your goal is conservation, you're still keeping them around at least. But I have here, this graph isn't accurate, but it's pretty much the least homozygous situation you can get. So then the hybrids, this is kind of the ultimate as far as outcrossing, because you're crossing between totally different species or different genera, even with orchids. We're very fortunate, I guess you could say, well, depending on your perspective. A lot of plant groups you can't cross between different genera, and in orchids you can, and that really, I mean, makes these really beautiful, lush hybrids. You know, we can have all these potential unique traits and really strong growing plants because of that. And that's just from a plant geek kind of thing. That's really cool too, because you know, species aren't supposed to cross, but yet in orchids we can cross different genera. So this is basically a horticultural pursuit. You get the novel phenotypes and durable plants out of hybrids. So I'll move into a couple examples. And from here on out, it doesn't really matter if you're talking about a species or a hybrid. The genetics or the traits that we'd be looking at, it's the same situation. You could use the same methods. And I'm going to start out with kind of a classic example. I don't know, there's other orchid history buffs that used to have poured through all the old orchids magazines or in books and heard of C.C. Hurst and his white Cattleyas, the C. Cattleyas and the R. Cattleyas. So, like I said early on, there's actually been very little work done on trying to understand the genetics, like orchid traits. A lot of individual breeders have done that and made observations, but to go from a more empirical approach and grow up hundreds or thousands of seedlings to actually do the research, that is kind of lacking. But well over 100 years ago, C.C. Hurst did some of this work. And we'll start with this example, Cattleya ludimaniana, a common, you know, everybody's probably familiar with Cattleya ludimaniana. So if we had a really nice white form and we crossed it to a pink form, what do you think we'd get? What color would the seedlings be? Very good. They'd most likely be pink because alba is recessive, the white flower is recessive. Very good. And this is what we'd say, like the genotype, or maybe you remember back from 8th grade, Punnett square. The C, the capital C, is the dominant trait, and it has color, it's pink. And then the little c is, so this is the genotype, or the genes, the alleles that the white Cattleya is carrying. This is what the pink Cattleya is carrying. All of them are pink. So we're going to do the same cross with different parents this time. And you get this. Any guesses? Right. The pink parent is likely heterozygous for flower color, meaning it has one of those little C's and one of the big C's. And when you cross it to the white, which has two little C's, you get this segregation, we call it, of 1-to-1, or 50% would be pink, 50% would be white. Now, this is oversimplified because, and I'm only representing 12 plants here, it might not actually be 6 and 6. It might more be like 3 and 9, or 5 and 7. You know, the numbers wouldn't be necessarily exact. So if we take any of those pink progeny from the previous two examples, and you could self them or sit across them, you would most likely get a ratio something like this, where you would get three colored plants for every single white plant. And that's because each parent has one allele that's white and one allele that's colored. And when you do the cross out, only one out of this combination is still white. Is that too that wasn't too in the weeds? Okay, then I put this up here.
One of the weeds.
So like I said, I'm not a biochemist, but I just kind of, this is off of a Wikipedia. This is the chemical, the component in the Cattleya that's giving it its pink color is an anthocyanin. You've probably heard that word before, anthocyanin. Anthocyanins are plant pigments. They help protect the plants against sun damage, usually in the foliage, because anthocyanins are also found in the foliage. but they also tend to give, there's other color classes too. Anthocyanins aren't the only one. You have the carotenoids that do yellows, but the anthocyanins tend to do your blues and pinks. And orchids use them to produce these colorful flowers to attract the insects. And what happens with the white plant is you have this long chain that starts all the way up here with the phenylalanine. All these steps have to happen to get down here to cyanidin, which is actually the pigment I believe that's in Cattleya's. All of those steps have to happen and have to happen correctly. If you have a mutation, and in almost every one of these steps has a gene or more than one gene actually controlling it. If you have a mutation in one allele of one of those genes, you can shut this pathway down. And I think the C refers to the anthocyanin synthase here, which is one of the last steps. That's an enzyme that actually takes this uncolored component and results in color being produced. So if you break that, the genes no longer work to make the anthocyanin synthase, you get white because there's just no color. So we'll mix it up a little bit. So I've given away some of my thunder here. If you know CC Hurst, you've done his work. So this hybrid crossing Ketleya triani with Ketleya leguminiana, I think it's called Princess. Somebody's recently remade Ketleya Princess, and it's with the ceruleiforms, the blue forms. But back in the day, when people are still figuring this out, you cross these two plants, and what do you think the progeny would be? Just guessing from what we talked so far, maybe. Would you get white plants?
You could get a surprising bunch of people instead.
Right. Probably going to get all pink, because it turns out that there's multiple mutations. What happens? There's more than one way to make a white Ketleya. Okay. That's what you get for, oh, I'm sorry, the cats. My husband, that's a joke from my husband. He was editing it for me before.
One of our breeders did that. They hybridized two white cymbidiums. Everything was pink. Everything.
Yeah, there's actually a lot of different groups. This is a really common situation in orchids. There's a lot of main groups. So there's actually two mutations that are going on here. The Ketleya leguminiana has one. The Ketleya triani has the other. So when you cross those two, they're each homozygous for their respective alba gene, but they're dominant homozygous for the other gene. And so when you cross it, you restore the pathways in both. And C.C. Hurst, over 100 years ago, discovered that. I was doing a little bit of reading about him the other night. He actually was working this out before Mendel's work was rediscovered. So it's kind of interesting that this was going on in orchids. So C.C. Hurst was in the UK. So, there could be a right here or right here, but when you put both of those together, they complement each other. So you get the restoration of the anthocyanin. And so this is actually what that cross would more look like, is you got two genes and the alleles of each gene. So the one white Ketleya was a C with a big R, little c with a big R, the other one was big C with a little r. And so all of them you can see now are heterozygous for that trait and restores color. Now, the situation is similar for, and I'm not going to go into this, they call it the P gene on semi-alba, and it's kind of a similar thing. It's a single gene that works to create the semi-alba phenotype or Ketleya as well. So what we've been talking about so far is genetics or genes or alleles, genetics that are qualitative, meaning it's a yes or no, it's a quality. If ever you grow out a bunch of seedlings and they can easily fit in either just 2 or 3 boxes, categories of phenotypes, we'd say that's qualitative or Mendelian. It follows Mendel's rules that he established all that time ago. But unfortunately, the vast majority of traits that we were worried about in orchids, like petal width and flower size and all that, are not Mendelian. It's not so simple. So here's a different example. And this is actually from a project I started a long time ago. And I crossed Phalanosis schilleriana with Phalanosis aphrodite to recreate Phalanosis leucorrhoda. Every single one of those plants, the progeny, all had this light pink color. These are the same plants. Unfortunately, I took this picture a long time ago in anticipation of eventually doing a talk, but my photo photography wasn't pretty great. So do you think that pink is dominant or white's dominant? A lot of time, you know,
Pink.
pink's dominant. How many people say pink's dominant? Does anybody say white's dominant?
No.
It's actually neither. So there's a couple things. We'll go into, it's actually different. But whenever you have a situation where the color is not, or the trait is not in either of the parents, there's different things. It could be either called codominance or incomplete dominance. And they get you to the same result, but they're two different modes. I won't go down with the differences necessarily, but I don't know if you ever, grow snapdragons. If you cross a white snapdragon and a red snapdragon, what color do you get? You get pink, because neither the white's dominant nor the red is dominant. It's a little different. That's actually literally like dilution, like we're mixing red paint with white paint, you get blue paint. Codominance is a little different, but that's exactly what's happening. But when I went on and I crossed I took two of those seedlings, and I picked, those aren't the actual seedlings, but I picked two of those seedlings and I crossed them. And I bloomed out like 80 of these things. And so this kind of represents the color distribution that I saw in all of those seedlings. So you can see there's everything from pretty much white to pink, pretty much close to the Schilleriana. So you have this grade that kind of goes from one parent to the other. So, I'll go back. So that's a quantitative trait, because there's multiple genes that are actually controlling the production of the pigments. And so anytime you get a distribution where you can't put them in neat little 2 or 3 boxes, but you get this more like distribution of color, that's a quantitative trait. And this is actually what most of the traits we're breeding on. So if we're interested in orchids follow, there's few examples like the color, but when people say, oh, I'm gonna cross, it's got a good gene. I've heard people say it's got like a good gene for full flowers or something like that. That's not necessarily true. It's got a bunch of genes that are helping get the full flowers. And another thing that I see sometimes in literature, and I see a lot of times on forums, is people say, oh, it's so dominant or it's so recessive. That's not a thing. There's actually not gradients of dominance or recessive. It either is or isn't. And I remember reading an article, I think it was actually like even Heather's an article, where he said he was talking about Ketleya breeding, and an alba popped up like 4 generations down, and he goes, that alba was so dominant. And I was like, no, it's just recessive. So, any questions before we move on? Any comments, any observations? Do people here make their own hybrids? I know some people probably do, no?
I think the people like you to make them buy some seeds.
That works too. That works too. So we'll go ahead and move on then to another topic that's near and dear to most orchidist hearts, the issue of polyploidy. So what is polyploidy? This is when an organism has more than 2 genomes. In animals, it's very rare, but in plants, it's very common. It's actually more common than you think. So a diploid plant has 1 copy of the genome from one parent and 1 from the other parent. But in this case, they have 2 or 1, or depending, 2 or more from at least one of the parents. The triploid has 3 copies, tetraploid 4, pentaploid 5, and so on. Usually the odd ploidies aren't fertile for various reasons. They don't make balanced gametes. They can't make an egg cell and a sperm cell that are viable. So usually only like things like diploid, tetraploid, hexaploid tend to be fertile. But what happens, I guess I'm jumping my slide, sorry. So, but how do we get polyploids? How do we have these situations where an organism, a plant, ends up with more than 1 genome? It actually happens naturally when there's mistakes during the process of making pollen and sperm, like I mentioned, and you get double the amount. Instead of going to 1 copy like you're supposed to, you get 2 copies or 3 copies or 4 copies. It's almost always just 2 copies though in that situation. And then if you have the pollen grain that does the same thing, you get usually triploids in this situation, but sometimes actual tetraploids. This can lead to new species because there are examples of orchids, as well as a lot of other plants that were there's a speciation event because some of them, there was an accidental tetraploid, and they kind of get cut off because they're no longer fertile with the diploid, so they kind of go on a march at their own separate rate now and evolve separately. For us, they usually happen accidentally in the tissue culture process. There's a process called endo-reduplication, and actually endo-reduplication happens in people too, like in our muscle cells and our heart cells, they have more than one set of genome, more than two. And sometimes it happens with plants where if you have a leaf cell that's finished dividing, it thought it was finished dividing, and so it actually duplicates its genome for various reasons naturally. And then you put it in tissue culture and that cell actually grows into new plants. Voila, you have a tetraploid. And so this happens actually, I think, quite frequently in the tissue culture process. And some people have I actually did a little experiment to try to take advantage of that. And you can make polyploid plants pretty easily without any chemicals. And that's kind of the main way that we get our potentially produced tetraploids or polyploids at least, is using toxins. Most of these things are toxic. They interrupt the cell function. Things like colchicine is kind of a common one. You may have heard people use that a lot. I don't think people use colchicine as much anymore as they use things like Arizalin or 2,4-D, which are herbicides and actually would normally kill the plant. But used in the correct way, you can actually stop the cell process from happening and have tetraploid cells at the end. I think Margieliantha Capra treats her plants and grows the seedlings. So you sometimes can see the plaster stem and hopefully pick out a good tetraploid from those. Or you can cross just two already existing polyploid parents. You cross a tetraploid with a tetraploid, you will get more tetraploid. So some pros and cons of polyploids. They usually have bigger flowers. I say usually. I have seen a lot of examples where that's not true. I've grown out a lot of different Phalaenopsis Aphrodite, amabilis, where the tetraploids or the purported tetraploids are actually a lot smaller than the diploids. So that's not always true, but it generally is true. Thicker tissue, that is almost always true. When you have all that extra DNA in there, it makes the cells bigger. It just physically makes the cells bigger. And so the tissue tends to be thicker, usually rounder. You get broader floral segments, you tend to, again, if you go that first picture, I guess I didn't stop long enough. I'll go back to it. This plant is a tetraploid, Phalaenopsis pulcherrima. This is also Phalaenopsis pulcherrima. And I put this in there because you can clearly see the rounder segments for sure. But this flower is actually larger, but this is the diploid. But this is kind of your classical tetraploid situation where you have rounder segments for sure. Sometimes bigger, sometimes more intense color, but here's, that's not necessarily the case. You can restore fertility in wide crosses sometimes with polyploidy. Don Wimber back, I think over in the UK not Don Wimber, Eric Young Orchid Foundation. I think they were doing work with like Phragmipediums where you could make the polyploid and it actually restores fertility. So that's one use for polyploids because if the parents are originally compatible, they have a very different number of chromosomes. And you make them tetraploid and cross them, you can actually have something that's fertile, which otherwise you wouldn't have. But it could also reduce fertility. Like I mentioned, if you get to a triploid situation, the triplets are usually sterile and aren't able to produce seed. You can kind of retain genetic diversity longer too, because when you have 4 copies of the genome, it takes longer to get them all back to homozygous. So if you're breeding like over a long term, it can retain that diversity a little longer. Blackberries are a really good example. They're tetraploids, and those crosses are crazy because there's just so many potential combinations in there. But the cons, there are cons to polyploids, and one of the big ones is they're slower growing, and some of them are excruciatingly slower growing. I've had some phals that were tetraploids that maybe bloomed every 3 or 4 years because it just took them that long to grow again. One of the biggest places, plants are out there photosynthesizing, making all the sugar to store energy. And one of the biggest consumption components of the process of that energy is replication of DNA. And so if you have twice as much DNA, it takes that much longer for the cell to get to where it needs to grow to actually to grow. It also is a big, where much of the nitrogen goes into the DNA. And so there's just a much greater demand on the plant than it's a polyploid. Sometimes you get distorted tissue and leaves. You see the Phragmipediums that are tetraploid, and you see the horns on the lips or on the petal edges and stuff like that. So sometimes, you can see things like that that aren't necessarily good. the tissue is often brittle too, 'cause it's so thick, it's really easy to break. And it complicates breeding, and we'll kind of get to that. So if those white Cattleyas that we were talking about were tetraploids, I had that nice little, neat little Punnett square that had just a few boxes. If you turn those into tetraploids, your CC whites and your RR whites, you have 256 different combinations of those genes, those alleles. And you'd still end up with 7 pink for every 1 white, the segregation ratio. But there was a very good reason that I chose diploid variants for my little Luca experiment, because I didn't want to have to grow out that many plants. I wanted to be able to see something with just 50. So if you're dealing with polyploids and you're interested in doing a little more kind of science geek kind of thing, they're much more difficult to work with. So this is an example that kind of blends both two of the things, the concepts we were talking about, this dominant recessive, or that we're seeing with the color, and the polyploid situation. Anybody remember when world-class Bigfoot first came around back in the early 90s? It was kind of cool. I think initially a lot of people kind of like, what's going on? It was registered by Carmela in 1990, and it got its JC in 1991. And it has a form of coloria, colorism, coloria, caloric flowers. What this is a situation where you have a plant that's normally zygomorphic. Orchids are zygomorphic. That means they have bilateral symmetry. They all have a top and a bottom. Whereas a daisy is not a great example. Maybe a petunia is a better example. It doesn't necessarily have a top and a bottom. They're actinomorphic, they're radial symmetry. And so in colorism or coloria, we have a return, in theory at least. I mean, I know it's not exact. We have this return of a more radial symmetrical flower because the lip is a petal. It's a modified petal in the orchids, but it kind of in this situation returns it to more like a petal instead of the normal shape of a lip. You can see the callus is still there a little bit. And this is different than most of the coloria you see in orchids. You usually see where the petals turn into lips. You see that in Phalanosis a lot. Splash petal Cattleya's, those are actually colorics, where the petals have the color from the lip coming in. So people have been breeding with this for a little while, and I was I saw it in the 90s, and I never bought one, but then I saw a really pretty, I was out judging on the East Coast, and I saw a plant on a table that was this was like early 2000s, I guess, very much like this flower here, and I just thought it was beautiful. So of course I had to buy it, and it turned out when I bought it, it was an unregistered seedling, but it had been since been really like the lip. And I had another plant that I really liked called Scarlet and Snow. I unfortunately have neither of these anymore, but I still have some of the progeny. Very much like this, a standard white with a red lip. And I thought it'd be interesting to see if I could you get like a red petal on there? So I crossed the two. And I registered it as Phalanosis ascota for this one. And this is kind of a representation of what I got for the cross, not the quantity. These are just lined up. But you see the diversity as far as the shape of the flowers, the fullness, the petal width, and things like that. I flowered out about 45 seedlings, and about 10 of them had the big lip trait. So 35 were normal. And so that's kind of close to a 3-to-1 ratio. And so we can kind of look and we can kind of deduce what's going on here, and that it's likely a single dominant trait following a Mendelian inheritance pattern. And here's the Punnett square. So the Scarlet and Snow, which doesn't have the big lip, just had the red lip, has a recessive allele for whatever the L gene is, the big lip gene. And this one just has one. And you see it's more complicated than the ones we showed in the Catleya because it's a tetraploid, and it's actually reduced a little bit for it. But this kind of shows the ratio that you get. And while that's not exactly 3-to-1, it's pretty close. And I'm fine just for my own purposes knowing that it worked. I think it's definitely a dominant situation. And it was interesting. So I'm going to change. Sorry. Was that
No, I like the color.
Oh, thank you. I'm going to change gears a little bit now and kind of talk about another kind of, I wouldn't call it necessarily a peeve or pet peeve, but it kind of is. But we'll start, I just talk about this hybrid. This is a little hybrid I made with Phalanosis Joyce Stewart. Joyce Stewart is Timothy Christopher crossed to Equestris. And then I crossed that back to Lindinii. So it has lots of Lindinii in the background, lots of Equestris. My goal was to make a little multifloral kind of Phalaenopsis with striped flowers that was very floriferous and hopefully bring in some fragrance. This Joyce Stewart it was awarded, and I call it Smelled the Roses because it was very, very strongly fragrant. And so I kind of think, well, for one seedling, I only ended up growing up 12, and only one looked like this. But when it was being judged, the program Orchid Pro that was being used, and it generates this file, and you can get this pie chart, and you can get this kind of pie chart on a lot of places. Like Orchid Roots does it. I think that's the other one. Orchid Wiz does it, and well, none of the judges that were on the team are probably here, but arguing that plant that has to be more full. It's got 12% Phalaphrodite in it. Do you really think it has 12% Phalaenopsis Aphrodite left? It probably doesn't. Definitely, we can say it has 50% Lindinii, but you really don't know that there's actually any genes, any alleles left from the from the Aphrodite, because Aphrodite is a big standard white. Selecting for smaller flowers in the stripes gives every time you select for like one parent or the other trait, you just kind of drag along more genes, more of the DNA from that plant, and you lose. I wasn't selecting for big, full white flowers. This was the parent actually. You see, it's a little bit fuller, but not all that much fuller. So this whole power of selection, whether it's natural or in our greenhouses, you can very, very much, get rid of it.
Just basically there's an unnatural selection against the big white.
Against the big white, right? I would have loved to keep the big, I was hoping, to be honest, I was hoping to have the white petals of but it didn't come through. But it was just interesting listening to, it has to, those petals should be 12. Actually, the comment was made, those petals should be 12% wider than that. And that's just so not true. So I think it's really, and I do hear hobbyists do the same thing, it's nice to have those programs and see, but you gotta know how to use those pie charts. They don't mean literally you have that much percentage of the plant left, because you're selecting away from that, you're gonna lose it. And different traits come from different sources too, and that's one thing I like about hybrids, is you can cross two things and things will pop up. I see this in my raspberry work all the time, but I see it in orchids as well. Things you didn't know were there, but when you put those genes together and they all play, you get different things than you didn't see. So this kind of goes along that argument. So these plants, they all originally had, their grandmother and grandfather. One was Schilleriana, if you remember, and one was Aphrodite. This plant, there's a random assortment, and that's something that Mendel actually worked out. I won't go into it, but it's the genes aren't necessarily linked to each other, or not very much anyway. So this plant here is actually probably closer, or has more DNA from the Aphrodite than this plant, which has more genes from the Schilleriana, just because it has the way that the genes are segregating out of this. Or if you remember, this was the F one, right? I or across the siblings, you got all those progeny out. Even the color change, the color kind of shows, or is representative of what's going on at the genetic level. And you are getting, if you continue to cross like these white ones together, you get rid of more and more of the Schilleriana. And if I had pictures of the leaves, you would have seen that, which I didn't take pictures of the leaves, unfortunately. These plants had leaves with that nice modeling from the Schilleriana, whereas these didn't. So it was impacting the whole plant, and you could totally go two different directions. And even though, according to the Orchid Register, this should be 50% Schilleriana. So it's not. So I mentioned about the 50%. This is the only time where you actually can know that the pie chart. If you have two species parents, you cross them together, they each represent 50% of the genome, more or less. And that's why when I said Lindinii, I can say there's 50% Lindinii, but I can't say there's 12% Aphrodite. I love primary hybrids because to me, they give me the satisfaction of seeing what the genes are doing together. But a lot of times they keep the grace or the charm of the species that go into them. They also often are a lot more easy to grow. So that's definitely definitely help. And I just have a few examples here of a hybrid. This is not registered yet, as far as I know. Arangus lutea alba, rotosticta specifically, crossed to Arangus macrocentra. Now the pictures of the macrocentra always make it tough because it looks like they're so much bigger. The flowers are really only half an inch wide, they're really short. It's called macrocentra, which means big spur. So that's where it gets the name from. But interestingly, when you cross it with the lutea alba, you get a much bigger flower. And now I know people, I've shown this to somebody and they're like, why would you make that cross? Well, I think it's really cool because the spur on these is much longer. So that comes through, which didn't come to the lutea alba rotosticta doesn't have. And to me, that's something that's really cool. I mean, there's no other arrays quite like that. And I definitely think all primaries are worth being attempted, even if they don't end up being valuable per se. But it's really cool too to see the orange color come through. And only in this situation can you kind of see the interplay of those two species. Now, the one next step would be, and that's something I will probably attempt to do, is to go ahead and sell for civities to kind of get that segregation out and see, can you get like a nice full flower like this with that nice long spur and maybe some fragrance. Lutea alba does have a fragrance, but not very strong, whereas the macrocentra is very, very fragrant. The hybrid was kind of intermediate with as far as fragrance. And the other thing, sorry for the fading flowers, I thought of taking this picture in the half, but when you make a primary hybrid, they seem to be very consistent usually. Very rarely do you see a primary hybrid where the plants are all over the place. They usually have very similar characteristics. So it's kind of, I know in Outcrossing people make hybrids a lot, or primary hybrids a lot because they're very repeatable and it's kind of cool. But this is kind of the equivalent of, your hybrid tomato in your garden. Even though they're from seed, they're all very similar, if not identical in the tomato case. But you see that uniformity a lot of times in primary hybrids too. Another one, unfortunately, I can't see the lineage. You can see it. Okay, it's coming through there. Habenaria, another group of plants I like to work with. I haven't been doing as much lately, but it's Habenaria. And this is Habenaria if you cross Robliniae with Carnea, Eucacoarea, which is a very, very nice hybrid. And again, you can see it's intermediate between the parents, and you can see, the color comes through, it's not quite the same. The shape is improved somewhat, but not I'd rather still have the Carnea shape. But you can definitely see the traits of both parent coming through. And another one, this one's a little maybe more obvious. Habenaria medusa, crossed with the Anthocyanin. This is what I think my favorite Habenaria hybrid. This actually was awarded at one of these judging here that goes on while your meetings are going on a long time ago. But you can see this was obvious. You get the lobules, I guess would be the name for these on the lid. Color comes through a lot more than I kind of expected. A very nice hybrid, primary hybrid. And then this is my favorite right now, my favorite hybrid, because I'll show you in a minute. But Callaway Borsovicii, which is the biggest Callaway, has huge flowers. And that's one of the reasons I like it. And Ludoviniana has some of the best shaped flowers. And these aren't the parents for the cross I'm going to show you, but just examples. But that makes Callaway permanent. And again, it's really cool. I go out in the greenhouse and I look at it, and what's coming through from each parent. You definitely got broader petals from the Ludoviniana. The flower size is huge. This is probably 8 inches. But that's coming from the Borsovicii. A little bit of the yellow in the lip again. Tends to be a Borsovicii trait. So definitely, if you like species, you want to grow something a little different, maybe some primary hybrids, because they're, it can be pretty cool. So that's the end of my talk. Any questions? I think I saw a couple of questions or maybe comments from the people looking up. So Pierre is asking about the difference between where the color is found. That was a road I decided not to go down because I didn't want to keep you guys here all night. But so there are different layers of tissue on a flora, and if you cut a petal like sideways, you'll see the epidermis, which is the skin, and then you'll see the mesophyll or the inner layers. And sometimes like in the case of the anthocyanins you see on the Catleya, they're generally restricted to that outer layer to the epidermis. Whereas if you have a yellow color, it tends to be more in interior layers. And so they overlay, and that's how we get a lot of our red colors, like the Phalanosis, the reds, that's because you're layering that pink over that yellow. And so if you should just talk about the difference or differentiate between the dominants and the different layers. Yeah, I mean, because they're independent, generally. The color, the colors that are the yellows that are usually on the interior of the cell are your carotenoids. Your fat soluble things or the anthocyanins are water soluble, and they're different parts of the cell. So they're generally independent. So you could have a situation where you have a red flower, across the red flower, and it's segregating for yellow, red, and pink, depending on how the things are being expressed in the different layers.
Yeah, he's coming from the perspective of Cymbidiums where they're trying to get orange and it's yellow with an overlay of red.
Yep. Yeah.
It's not really orange.
Well, and that's the same way that with Phalaenopsis, the oranges and reds, it's the same way. And even I think in some of those really nice dark red Cattleyas, I think that's what you've got going on as well, is layering the two together. Does it matter that it's the effect's the same, right? I mean, if you look at it, it's orange, then I guess you've accomplished it, but if it's not really orange, you're right, it's not orange pigment, that is for sure. Did I totally confuse everybody, bore everybody? I hope you at least learned something or maybe and I forgot to mention, point out, but some of you saw these golden raspberries, they actually have a little bit of red over it. And just to show the anthocyanin synthase, the ANS gene that I was pointing out, raspberries have the same gene, most plants have the same gene. And in raspberry, this is a mutation of the anthocyanin synthase gene because the endocarp or the mesocarp, the inner tissue, has the red gene there, but it actually is lacking the anthocyanins being expressed in the middle, so you get a golden color instead. But that same mutation isn't impacting the skin, so you get that little bit of. But it's the same exact gene. I mean, it's probably a little different sequence-wise between the Catleya and the rubus because there's a lot of evolution between the two. But it's essentially the same gene doing the same thing in a different plant. So I just want to point that out.
Thank you.
While Lynn's setting up, should we ready for show and tell? Lynn?
I'm getting there.
Okay.
Let's start.
Okay, what do you think?
Nothing yet.
Nothing yet.
Do you see my first slide? No, not yet.
No, we can you.
Really.
You see the dog bed.
Okay, I quick shared screen. Now we're going to go click on slideshow. Beginning. What do you see?
Nothing
yet, Lynn. Okay.
I see a dog who prefers a chair to his bed.
Yeah, there's Sasha.
Oh, wouldn't you?
There she goes.
She's funny. She's smart.
Okay, so I click share screen right here. Share the screen too.
It's shared.
No problem.
There you go. Hello.
Show your notes, Lynn, still.
Okay.
Hit the enter icon. Oh, you did.
Perfect.
Okay, do you see it without the notes? Thank you, Tom. This is the last time I'll struggle through this, I hope.
Stay, Lynn, stay.
There goes Sasha. Okay, so we have a shorter show and tell tonight. We only have 32 orchids, but they're no less beautiful, and here we go. Tyler Albrecht starts us off with his Paphiopedilum Charlesworthii. Charles Worth was a 19th century British orchid nursery. It was not an individual. This is a species found in Burma, which is now Myanmar, and found in 1895, growing in shade with their roots clinging to the crevices in rocks from about 4,000 to 5,200 feet. So it's a relatively cool grower, prefers shaded to dappled light. Plant breeders really love this large, glossy pink dorsal sepal, and they often use it in their hybrids, hoping to achieve a clear pink dorsal like this, but rarely successful, according to Harold Kupowitz, who says that mating this solid pink dorsal with a standard complex hybrid usually results in a white background with pale purple spots. So this is Charlesworthii as a medium-sized paph. The flower is over 3 inches, just lovely. This is Tyler's Paphiopedilum adductum rus carpals. This species is from the Philippines and was first collected in 1950, so it's a relative newcomer. It's obviously a multifloral. It's been referred to as the Darth Vader of the orchid world because of the dark color and that hooded dorsal, giving it kind of an ominous look. This is considered a difficult paph to grow, and Tyler's doing a really good job. He has 2 flowers and 1 or 2 more buds, I can't tell which. The flowers are about 3 inches and up to about 20 inches long with the graceful lateral sepals. I think this is our newsletter editor's debut on virtual show and tell. Eugene Kim, glad you're joining us. She shows us her Aranthes ramosa, which is a charming little species found only on the African island of Madagascar. As an epiphyte, it grows on trees in mossy forests at about 4,500 feet of elevation in quite low light. So that is the tip for how to grow it in cultivation. The nearly translucent green flowers are about 3 inches wide. They're carried sequentially on an inflorescence, which may reach up to 5 feet on a mature plant. The inflorescence can take months and months to form. It should not be removed until it's clear that it's dead and brown, because it can rebloom off the same inflorescence. So intermediate or household temperatures in low light with good air movement, and behold the beautiful result. Andrea Lodate shows us Dendrobium Patriot Beauty. This is a hybrid by Roy Tokunaga of H&R Orchids in Hawaii. We miss seeing Roy at our shows, but his hybrids, there are over 350 of them, continue to be available in the marketplace. This one has just 3 species in its background. And as you can see, it's 50% Dendrobium spectabile, which is the one with the wild and crazy shape. The result of the cross is this really pleasing rose flower with deep rose stripes and a white picotee. Like most of Roy's Dendrobium hybrids, this is a great windowsill plant in bright indirect light. Do reduce the water in winter when it's not actively growing. Andrea was thrilled to come home from vacation to find this in bloom. Andrea's Masdevallia barlayana, named for Mr. Barla, who was a Brazilian orchid enthusiast in the 1800s. It's native to Peru near Machu Picchu, much like Masdevallia vechiana. It grows at a really high altitude, so it's a cool grower, which will thrive outdoors in San Francisco's cool, foggy climate, which is where Andrea grows it. The crimson flowers are about 1 inch wide, about 1.5 inches long, including the caudae or tails, and it looks like it's thriving on Andrea's deck. Dale Martin always sends me photos of 8 or 9 of his beauties, and he makes me choose. So this month, we're going to focus on his bulbapillums. The first one is Bulbapillum Macrobulbum, a smelly one, but he says it's not as stinky as some. This is found in New Guinea on moss-covered trees or rocks at low elevation. So this is a warm grower, needs partial shade and good humidity. A single leaf arises from each very large pseudobulb, and the leaves can be up to 3 feet long by 4 inches wide. So this is not so great for you windowsill growers, but it's a spectacular specimen and a great photo showing all of the intricate parts and colors which work together to beckon the pollinator to come hither. That is gorgeous. Dale purchased this with the label Bulbapillum yasnae, but he believes it's actually Bulbapillum plumatum, which has much longer sepals like we see here, 4-inch flowers per inflorescence. This species is found in the Philippines and in Sumatra, in swampy forests at low elevation. So it's a warm grower, should be kept moist and with high humidity year round. It looks like Dale is growing this in sphagnum to retain moisture and in a hanging basket so the flowers can hang down gracefully. This is lovely. This is Dale's Bulbapillum adorable upwind. As you can see in the top right, Bill Toms, he's a great grower and hybridizer from Florida. He's been busy creating bulbo crosses, many of which he has named adorable. So his wife's name is Doris. So you can see where adorable comes from. And this is adorable upwind. This is a primary hybrid of 2 bulbo species, Basacetum and Echinolabium, from the Philippines and Sumatra respectively. They're both warm growers. That elongated lip is hinged, as are all or nearly all bulbos. So that hinge means that when the pollinator lands on it, possibly a fly, lands on top of that lip, the lip tilts him up toward the inside, so the pollinia comes off on his back before he buzzes onto the next flower and pollinates it. This is Dale's Bulbapillum longicepilum. If you love weird and unusual orchids, as you can see, bulbos are for you. Bulbapillum is the largest genus of orchids with over 1,500 species, and it's also the most geographically diverse. It's found in all tropical areas on Earth and is quite diverse in its flower morphology as well. Longicepilum means with long sepals, about 3.5 inches long, and it is just the sepals which we see in this photo of the flower. The petals and the lip are minute and they're hidden up inside the top of that flower. This species is found in southeastern New Guinea as a miniature-sized, warm-growing epiphyte. It looks stealthy to me. What a nicely grown collection of bulbos, Dale. This is Tom Pickford's Ketleya tenuis. This is a Brazilian cool to warm growing species found at elevations up to about 5,000 feet. It grows on upright tree trunks near the top of the tree, with the roots running several feet along the trunk. It's sheltered from the sun by the foliage, so it needs bright filtered or indirect light. The inflorescences then grow very long to push the flowers up into the light, so they're more easily found by the pollinating insects. So clever. The waxy, fragrant flowers are about 3 inches. This is a bifoliate, as we can see in Tom's photo on the left. So great care needs to be taken to repot only when absolutely necessary, and then only when new roots are starting. It needs a dry rest for several winter months. Tenuis is similar to Ketleya bicolor, which I just dropped in on the right-hand side. It's another Brazilian bifoliate species, but we can see the difference in the flower and especially in the lip. Tom's Ketleya Good Days 90s. This is an unawarded primary hybrid of the 2 species shown, Ketleya walkeriana by Laelia rubescens. The parent Laelia rubescens is a small rupiculous orchid, and it brings down the size of this plant, so it could be a great windowsill grower with bright indirect light and intermediate temperatures. Tom's has been blooming for 2 or 3 weeks already, and the flowers are still fresh. This plant needs to dry out between waterings. And Tom's Rhyncholaelia Ketleya Cynthia, another lovely walkeriana primary hybrid. This time walkeriana is crossed with Brassivola digbyana, which is the one with a wild and crazy fringed lip that you see on the right, bottom right. The resulting hybrid looks very, very much like the walkeriana parent, but with a different midlobe or lower portion of the lip. This is a hybrid that was registered in Britain in the early 20th century, but the cross was apparently remade by Fred Stewart Orchids in San Gabriel, California, because all of the early awards are to his clones in the 1980s, and all but one of the AOS awards has been to growers in California. It's a beauty. Dave Hermeyer shows us Ketleya intermediate pine knot. Intermediate is another Brazilian species. This one's found in Atlantic coastal forests and trees, rocks, and in the sand dunes, actually. It's named intermediate because the flowers were considered intermediate in size between previously described Brazilian unifoliate cat labiata and the bifoliate cat lodogezii. There are short and tall populations of Ketleya intermediate. Short ones are up to about 10 inches, and the taller ones are up to about 20 inches in height. And Dave has one of the more compact forms here, growing beautifully on a mount. You can identify an intermedia even when it's not in bloom, because the base of the leaves are serrated. You can easily see and feel the bumpy serrations. There are more than 100 recognized clones or color forms of intermedia in Brazil, ranging from this alba form to deep pink magenta lips and petals. This is Dave's Laelia Diana, though the taxonomists have changed the name of this to Ketleya by Callaway. This is another beautiful Brazilian species. It's small, it's only about 6 or 7 inches tall with a short inflorescence and a relatively large flower up to about 3 inches, with a beautiful magenta to purple keels or raised stripes inside the lip. Those are there to beckon the pollinator to come on inside for a good time. This can be grown intermediate to cool with winter night temperatures into the 40s, as long as you keep it much drier during cold spells. This is a possibility for windowsill growers as well. This is Dave's Yamadara Clarissa Caguan. Yamadara is a man-made quadrigeneric orchid hybrid composed of Racevola by Ketleya, by Epidendrum by Laelia. And here's one of the results, Clarissa Caguan. Interesting that it opens purple and fades to magenta as the flower ages, and that lip really steals the show. Dave grows all of his mounted orchids really well. And this one can also be grown potted. It likes very bright indirect light, warm to intermediate temperatures, another lovely candidate for the windowsill grower. Kay Klum shows us Rodriguesiopsis Eleutherocephala. As Kay says, the name is bigger than the plant, which maybe is, she has her ruler there, is maybe 6 inches from top to bottom. And the little green and white flower is about 1/2 inch, marked by tiny magenta dots and blotches. The genus name sounds like a man-made hybrid, Rodriguesiopsis, but actually it's a genus found in central Brazil with just 2 epiphytic species in the genus. It's similar to Gumza, but apparently with enough difference in the lip morphology that the geneticists have not yet lumped them together. Just wait. Kay grows this in the intermediate greenhouse. This is Kay's Epidendrum Hugo-Medinae, a species from Ecuador, named after a fine tour guide and botanist, Hugo Medinae. It grows at around 5,000 feet of elevation, so Kay grows it in the intermediate greenhouse. This is a reed stem epi, blooming at the apex of the cane, and according to J. Fall, the inflorescence can rebloom for several years, and the 1/2 inch white flowers are sweetly fragrant at about noontime. This is Kay's Aranthes neoperierii, an epiphytic species found in mossy evergreen forests in the eastern part of the island of Madagascar at elevations around 1,000 meters, about 3,500 feet. They prefer shady, humid, well-watered conditions and even fertilizer year-round. The inflorescences can take months to form, just like Eugene's ramosa. So they should not be removed until it's obvious that the inflorescence is dead, because they can rebloom off the same spike. Kay brought this back from Malala and Madagascar about 10 years ago, and it grows in the intermediate greenhouse. This is Kay's Bulbapillum odoratissimum, which Kay says, despite the name, has very little odor. It's found in Southeast Asia in evergreen lowland forests and obviously has a creeping growth habit. The flowers are about 1/4 inch wide and are in a cluster which arises at the base of a pseudobulb. It likes partial shade, warm temperatures, and regular water and humidity. Roberta Fox shows us two stanhopas, Wardii and Oculata. From Mexico to Brazil, this genus is hard to beat for its size and beautifully odd complexity, and there's 67 different ways or species that nature has come up with presenting stanhopa to us. Wardii on the left has a sweet, musky fragrance exuded as the flowers open with a pop. Each flower lasts only a few days, but with multiple inflorescences that do not all open at once. The show lasted almost 2 weeks for Roberta. And on the right, Stanhopa oculata, the name oculata referring to the eyes, the circular spots characteristic of this species. Both of these grow at elevations below 6,000 feet, so both should be grown in a wire basket, as we see here, because the inflorescence hangs pendant from below the plant. Roberta grows them on her patio in coastal Southern California, which probably translates to intermediate or probably not windowsill. It's too big here in Northern California. And Roberta's Dendrobium smiley. Dendrobium is the second most common orchid genus in cultivation after the Ketleyas, and it is wonderfully diverse. And this is a good example of the diversity from what you generally think of as a dendrobium. This is native to New Guinea and Queensland, near the edge of rainforests at low elevations. So Roberta grows it all year in her greenhouse and keeps it moist all year. It blooms on canes from prior years, and each cane may bloom more than once. In Australia, it's known as the bottle brush orchid. This is Roberta's Acantharia, or now Pleurophthalus strupifolia. The thick succulent leaves indicate that it probably experiences some dry periods, and the leaves act as kind of a storage unit, like pseudobulbs on plants with pseudobulbs do. Excuse me, it's a species from southern Brazil and Peru. Although it was first described by Lindley in the 1830s, very little information is available on this, but it likely grows at fairly high elevations, as it does well on Roberta's Southern California patio throughout the cold winter. Like most pleuros, you can see that the inflorescence arises from the top of the leaf. You can see that on the left-hand photo. This is Roberta's Ketleya quinquecolor. Quinque means having 5, but I'm not sure what this flower has 5 of, but it is lovely. This is a primary hybrid of Ketleya clandii by Cat Forbesii. Ketleya clandii gives it the spots and the color, as well as a light fragrance. It blooms several times a year and is very cold tolerant, and these are characteristics inherited from the Ketleya Forbesii parent. So unlike a clandii, it does not need warmth, so it has the best of both parents. Roberta says this is an extremely vigorous plant. This is a beautiful blooming of it. Susan Anderson shows us Neopathia Superior Eye, named after the same 20th century French orchid botanist as Kay's Aranthes, which we saw a few slides ago, Mr. Perrier, probably the water guy. There are 5 species of Neopathia found in Madagascar and Comoros. And this one must be very rare in cultivation because Ron Parsons spotted this in Susan's greenhouse and took a zillion photos of it. The flower is about 1 inch wide with a 5-inch spur or nectary. The first AOS award on this species was to a local grower who many of you knew, Lil Severin. It was awarded in 1978, and the award description described the spur as being in the shape of a shepherd's staff, and the lip reminiscent of a medieval halberd, which I had to look up. Our descriptions are nowhere near as colorful today. Susan grows this in her intermediate greenhouse in filtered light. Susan's Chrysocentron marrellianum is found only in Sabah, Borneo, in lower to upper montane forests and riverine forests in crowns of large mossy trees at elevations from 3,600 to nearly 10,000 feet. So it can grow cool to cool intermediate and bright indirect light. This is a small monopodial epiphyte, grows well mounted, as Susan's is here. And Susan's Angricum dallii is a charming little species found in Madagascar in dry forests, elevations around 3,300 feet. So it can grow cool to intermediate. It's best mounted or in a wood slat basket grown with high humidity, moderate shade, and ample water while in growth, and less water when it's not in growth. But it should never be allowed to dry out for long periods. Gloria Justin is new to show and tell, and we welcome her. Unfortunately, the names of Gloria's plants did not come through with her slides, but this looks to me like a lovely little Macroclinium, which is a New World genus with 42 species from Mexico to South America. They're typically twig epiphytes, and they have a reputation for being not very long-lived in cultivation. So this is a really nice, nice blooming. This is Gloria's Phalaenopsis species, or maybe a primary hybrid. Sorry, we don't have a name. And Gloria's Phragmipedium something. Again, I'm sorry we don't have the names of her plants. This is my tiny Margieliantha Capra, which I purchased from Afri Orchids at the POE pre-sale in February. This is a miniature, cool growing epiphyte found in South Africa at about 6,000 feet of elevation. So I grow it in my cool greenhouse with good humidity and good air movement 24/7 in dappled light. The flowers are sparkly white. They're less than ½ an inch, and they're translucent with a translucent nectary. This is my Arpophilum Laxiflorum, which I got from Asuka at SFOS's 2018 holiday party. And I always like to think of Asuka when I look at this plant. Asuka had been awarded a cultural award by Pac Central on his mother plant, which had 1,500 flowers on 22 inflorescences, and he grew it outdoors. I have not come close to that floriferousness, but if you love Arpophilums, this one is small. It's only about 16 inches tall, or about 1/4 the size of the typical Arpophilum gigantea. Laxiflorum is native to Mexican cloud forests at elevations over 8,000 feet, so it's a good outdoor candidate in the Bay Area. And this is my Dendrobium helwigianum. It's a New Guinea native in the Oxyglossum section of Dendrobium. It's a small, cool to cold growing species found on horizontal branches in mist forests. So I grow it in the cool greenhouse in fairly bright indirect light with good humidity, and I water it twice a week. The flowers are about 1 inch and they last for 3 months or more. Unbelievably, they look very delicate, but they're very long lasting. And I got no photos for the Pet Orchid Parade this month. So here is our now year-old English Springer Spaniel, who you see moseying around behind me. This is Sasha, and I'm sure she's dreaming about orchids. So that's all for tonight, and thank you very much.
She
Great
wants
job,
orchid
Lynn.
cookies.
Lynn, thank you so much for all of your effort.
Great job.
Thank you. I've enjoyed doing it.
All right.
You're gonna miss Sasha.
So Lynn's just taking off. She's going to be traveling next month. So we're going to be looking for someone new to do show and tell next month. So we have to think back to what we used to do before Lynn did this. And then make plans for next year, because on December, as I said, we're going to just do, well, I guess we could have show and tell in the meeting. Sorry.
Yes.
So I'm going to switch to try to do it on my phone now for the ones in the room.
And then we can just watch it up here.
Yeah.
Right.
So,
Sure. Well, if we're gonna watch it up here, just leave the lights where they are.
Yeah, they're probably
I have to figure out how to do this. So you're going to have to pin me, right?
She has to stop sharing. And that just makes it harder to see the screen.
Yeah.
It probably best to turn those lights off so we can actually see the screen.
Yeah. Can you pin my video? Or
Keep going. Thank you.
I don't see.
I don't see him at all.
Yeah. I
Oh,
don't
I'm listed as.
Oh, here you are.
There you are.
It was,
Oh, yeah, there we go.
All right.
You want to pin that FFW, whatever.
All right.
So let's see, we'll start with, I think these are brought in by Mitch Schneider. Let's see, this is an area, Reginaria carnea by Callaway. By the way, Mitch is also going to be a vendor here week with your New World Orchid, so you can see his tag here. He also brings in started off on, it was also I don't know if you guys can you hear me. This is a Don Quixote Harry Baldwin by Teak Shine.
Jeff, you're muted.
We can't hear anything.
Oh,
All right, can you hear me now? Whoa.
Okay, echo, echo. Dennis, fix the echo.
Now? No.
Super echo. Fix the echo.
There we go. Can you hear me now?
Yep.
Okay. All right. So this is Bill Weaver's Ketleya Mott Spot by Long Life Spot. This is his Packy, Band of Packy. This is Bill's. I can't read that. Something Bumblebee SVO by Athena Elizabeth. What is that?
It's a zygopedalum cross.
Zygopedalum cross. All right.
Why don't we do this, bring it over here, then you can look behind me and see what's on the screen.
I think this belongs to Mitch again. This is Odontoglossum Summit. The Vanda in the back is mine. This is from, I think I got this from Moats. And it's called Coltana New Release Maui. And I didn't do these either. So this one, these are Vanda Walkeriana by Intermedia. Cerula, I got this from Dave Hermeyer. And then there's Prostechia, another one that's from, can't find the tag. That's from Mitch. Then there's a Miltonia. This is from Jan Anderson, Regnellii. These are very delicate. I don't know if you can see these. Mosquito orchid, okay.
Epidendrum diffusum from Jan. Then K brings in, this is Volusiella maudii.
That's the little one that's hanging on it.
Oh, that's this guy?
Handle there.
Very little.
Yellow.
Volusiella.
From Madagascar. Well,
It's-
actually, no, it's from Africa, up and down Africa.
Ah, yes, this is the Proseccia, the one over here. Species. All right, and then K brings in, this is a dark Maxillaria schunkiana.
From the hill.
Oh, wait, I lost the, can you see it? It's like red, black. It's so dark. All right, and then these long Oberonia.
The big one is Obisa, and the little one is Mondusima. The Obisa has a dozen spikes on it. Yeah.
This is the little one. What's this? And then there's a nice Vanda over here. Oh, I think this is also Jan, right? Jan Anderson brings in Vanda cristata versus Suarez. Suaves. And then this is from Japheth Co. Ketleya, is it Longipes? I don't know how to pronounce that. Soft touch. And another one. Cool. All right. Is that it? Anyone else? Let me stop.
You stay on.
All right. Okay, so I think this is it. We're going to do the raffle in the room, and we'll see everyone. There's a show board meeting with the show committee meeting at Mishi's place and our online next Monday at 7. And the next meeting, we have Fred Clark here in November 1st. So till then,